Hello everyone, it's Benito here. Now today is going to be a completely different video to normal. It's probably the closest thing on my channel yet, at least, to a vlog. And to be honest, I don't know whether that's a cringy thing to say or something which I should be proud of. But I thought I'd finally spend some time talking specifically about my scientific research, and all of that goes on in that building there. That is the Life Sciences building here at the University of Bristol. Without further ado, let's get in there and let's see what the hell I get up to. Come on, get with me. Right, so this is my office. This is where I spend a lot of my time, but obviously it's in the lab where all the exciting stuff happens. So that's where I'm going to take you now, and it's where I'm going to explain what the hell I'm doing as part of my PhD. But the clue is here on the walls of my desk, because these are the animals which I'm studying. Right, let's go up to the lab. Right, so we're now entering laboratory territory. Now, this room, so you may not look like a typical lab, it's just full of computers, but actually a lot of very, very important work related to my PhD goes on in this room, and that's because these are very special computers, of course. These are 3D imaging computers, so they're going to help me through many of the analysis aspects of my PhD. But to understand why, I'm going to have to tell you a bit of background. So let's sit down and I'll give you the goss. Now, any good biological question should start in the field, really, not in the lab, because ultimately we're interested in how nature works, how it functions, and as an evolutionary biologist, I'm interested in how organisms are adapted to the environment in which they live. Now, if you've been following my Instagram account, @BenitoExplained recently, you'd have got a bit of an idea of what I'm talking about, because during the Christmas period I took you to my local woodland and described how forests are quite heterogeneous habitats. By that I mean, despite woodlands and forests being habitats in their own right, within them there's all sorts of microclimates, all sorts of different microhabitats. And that means loads of different species of animals, plants and fungi can live there. Now, if the forests outside the back of my house have loads of microhabitats, imagine the number of microhabitats in the Amazon rainforest, the most biodiverse area of the planet. And it is there where the study organisms for my PhD live. And they are butterflies, more specifically a tribe of butterflies called the Ithomini. Now, the Ithomini are extremely diverse, but what I'm interested in is their evolutionary interactions with this tropical rainforest environment. One of the special features of these Ithomini butterflies is that they mimic each other. Now, I don't want to give you too much detail too soon, but essentially, different mimicry patterns occupy different microhabitats in these Amazonian rainforests. So what I'm interested in is how that impacts the sensory systems, the eyes, the brains, the genes of these butterflies. And here in this imaging lab, that's where I start to look at that a little bit. So what you're looking at here is a butterfly brain. You see, to understand what sensory information a butterfly is taking in from the world, then you could do a lot worse really than looking at the brain, because inside the brain there are different regions, we call them different neuropils, that encode different information. They process different types of information. So these bulbous things protruding from the sides are what we call the optic lobes, and they process light, visual information coming from the eye. So what I'm going to be doing is looking at this huge community of butterflies of around 60 species in this Amazonian rainforest in eastern Ecuador, and then comparing the brain volumetrics of the different species and seeing how that relates to the microhabitats, the environments in which these butterflies a living. But we also have another lab which is just along the corridor from here where all the not so computational stuff takes place. I'm just going to take you over there now so you can see what it's like. Right, so this is my lab which I share with all the other members of my lab groups. There's about 6 of us. Haven't been doing an awful lot in here at the moment, but I'll no doubt be showing you some stuff as the weeks, months and years go on. But the really exciting bit in this lab at the moment is down here. That yellow label says Benito's samples. No, it's not what you think. I'm talking about butterfly brain samples. That's what's inside this box here. Inside this box here are about 40 tiny butterfly brains. So inside each one of these little containers is a butterfly brain, which I've since realised is going to be almost impossible to film, but I can just about see it at the bottom of that little container here. Can you? Unlikely, but there you go, very exciting. That just gives you a scale. I mean, it's all very well, seeing it on the computer screen, but it really puts things into perspective when you look inside one of these tubes and you really get to see how small these butterfly brains are. Crazy. Right, I think that's more than enough for one day, but thank you for watching this video. No doubt I'll be giving you more updates and more information on my research as I progress through my PhD. But if you enjoyed the video, don't forget to give me a big thumbs up down below, subscribe, of course, and also go and follow my @BenitoExplains Instagram account, where you'll receive all sorts of exclusive wildlife and science content. For the meantime, I'll see you next time, folks.